What we observed in Maya was just as in Pollux, the dara-lendex combination appears to be well tolerated. There is a higher incidence of neutropenia, as well as increased incidence of infections, particularly upper respiratory infections. But it does not impede in the survival outcomes. Um, What's also important to note in my experience is that the lenalidomide dosing was modified more frequently on the experimental arm. I believe somewhere close to 76% of the planned dosing of lenalidomide was delivered in the experimental arm compared to the standard of care arm. And this observation was also seen in Pollux and has also been actually observed with pomalidomide in my experience. From a clinical standpoint, again, I'm simply stating this from my own experience. I suspect that for the most part, when we're combining dara with the immunomodulatory drugs, we will likely be dose reducing patients fairly quickly in this first cycle or two. The GRIFFIN trial is a phase 1 slash randomized phase 2 trial that began here in the United States roughly 2 years ago. It had a safety phase 1 run-in, which included 16 patients. Essentially, the idea was, what are the safety implications of combining daratumumab with RVD as part of induction and consolidation in transplant-eligible patients. And these results were presented at ASH in December of 2018, showing that there were no major safety implications in terms of increased risk of infections or ability to collect stem cells in patients or post-transplant recovery of neutrophil counts as well as platelet counts. I think these are very important pieces of information that were necessary to be generated in the safety run-in part of the trial before the randomized portion could begin. The randomized portion of the trial consists of 200 patients. The trial is fully enrolled. It randomized patients to the standard of care RVD induction, stem cell transplant, followed by consolidation and then maintenance with lenalidomide, whereas the experimental arm added daratumumab to each of these stages of treatment in terms of induction and consolidation, followed by maintenance. So we're just waiting for the data to mature and inform us on the implications of incorporating daratumumab into the frontline setting for transplant-eligible patients. What we know right now, it's a safe strategy. We're simply looking for the efficacy signal. In terms of the efficacy results of the phase 1 run-in portion of the GRIFFIN trial, the VGPR or better rates were 63%, and these included complete responses and stringent complete responses. MRD negativity was achieved in 8 out of 16 patients. It's important to note that the median follow-up was about 15.6 months, and we know that responses do deepen on over time. So hopefully in an update, we'll see that these responses continue to deepen with higher rates of MRD negativity. All the subjects in the phase 1 run-in portion of the GRIFFIN trial were able to proceed with stem cell transplantation, and 15 of the 16 patients remained progression-free at the time the study was being reported. I think from a safety analysis standpoint in the phase 1 portion on those 16 patients on GRIFFIN, the most important thing to note is the cytopenias that were observed. They're consistent with what we see with RVD. They were seen in 10 out of 16 patients. The infusion-related reactions are probably important to note. They were seen in 31% of the patients, and vast majority of those were patients who were receiving daratumumab for the first time. These rates are certainly lower than what have been reported for other frontline or even early relapse trials, and probably represent the use of montelukast as a pre-medication for these patients. The grade 3 or higher adverse events on the safety run-in portion of GRIFFIN, the most common ones included hematologic adverse events, febrile neutropenia, as well as pneumonia episodes. But none of these adverse events actually led to the discontinuation of subjects from the study.